Welcome grade 11 learners to term 1 revision and consolidation. Guys, I know that you're into the end of the term, busy with exams, probably revising. And what best way to do this? So let's get involved with an interactive lesson. I've got some questions here on the work that we've done since the start of the term. A quick recap on the term's work. So guys, we started off the year with looking at microorganisms. We looked at the diversity of microorganisms. We looked at bacteria, viruses, protists, and we went on to fungi. And then we went on to the study of invertebrates and we looked at invertebrates. We also spent some time at school looking at immunity and how immunity is acquired. So guys, this lesson is essentially a collaboration of what you've done this term, a good revision for you guys to use as part of your preparation for the end of term assessments. So let's get straight into it. So we start off with a whole lot of multiple choice questions. Guys, I'm going to read through these questions for you. And I'm going to give you a little moment once we get through all the multiple choice questions for you and your team of friends to work through this. And then when we do get back, we're going to look at the potential possible answers for these. So again, 1.1. All fungi are A, parasitic, B, pathogens of plants, animals, or bacteria, C, able to photosynthesize, and D, heterotrophic. Next question. Malaria is an infectious disease caused by a single-celled organism carried by female mosquitoes. The mosquito is the A, pathogen, B, vector, C, agent, D, disease. Question 1.3. How does immunization against diseases such as diphtheria and polio limit the spread of these infectious diseases? A. Immunization kills the relevant pathogens. B. Immunization suppresses or reduces the immune system response and associated inflammation. C. Immunization strengthens first-line defense barriers and prevents the entry of relevant pathogens into the body. And D, immunization reduces the multiplication of the relevant pathogens in immunized host, and this reduces the chance of other people becoming infected. Next question, 1.4. The relationship between rhizobium and leguminous plants, in brackets legumes, represent an example of A, parasitism between rhizobium brings about the decay of the legume when it dies, thereby returning the nutrients to the soil, or B, mutualism because rhizobium denitrifies the legume and returns nitrogen gas to the atmosphere. C, parasitism between rhizobium denitrifies the legume and returns nitrogen gas to the atmosphere. And D, mutualism benefits rhizobium because, I'm going to reread that again, guys. Mutualism because rhizobium provides usable nitrogen-free legume and legumes provide rhizobium with organic nutrients. So guys, we've looked at 4 multiple choice questions. I'm going to give you guys about 2 minutes to read through these questions and then to work through these solutions. When we get back at the end of 2 minutes, we'll discuss some of these answers. So go have a good discussion. Welcome back, guys. I know that there was lots of reading, so I decided I'll give you an extra minute so that you can work through those and have a good discussion with your friends. So let's look at these questions. Let's look at the answers, and remember that you've got to read, underline the important keywords, look at the various options before you decide on an answer. I think it's very important. Often in multiple choice questions, the possible answers are quite confusing, and you might easily tend to read, and what is obvious, you might tend to do that. But read the options carefully, weigh the options out, spend some time on this so that you know that the choice of your answer is definitely your choice and not out of confusion. So the question again, all fungi are, and guys, if you go back to the section that we did, you know that fungi are, they're not capable of photosynthesis, and hence we know that they are not capable of photosynthesis. They are pathogens of plants and animals, and guys, we know that that's not true because they often grow independently. We do find pathogens on animals, but they're not necessarily pathogens. These fungi grow as parasites on them. All are not parasitic, and hence that's not correct, but we do have them being heterotrophic, and that means they often rely on other organisms for their source of nutrients, where they secrete an enzyme and they live off the medium or the substrate by absorbing the nutrients. So well done to those of you that got it right. Let's move on to 1.2. Malaria is an infectious disease caused by a single cell organism carried by female mosquitoes. The mosquito is and guys, if you recollect, we definitely did the life cycle of malaria, and we looked at that the malaria has two hosts. It has a human host and it has a vector, which is the mosquito, and the mosquito is not affected by it in any way. The Anopheles mosquito is not affected by it in any way, and hence we will not regard the mosquito as the pathogen. So it's not the pathogen, it definitely is not the disease, it's not the agent, but it is the vector that actually transmits the disease from the host, which is a human, to an uninfected mosquito, and then that life cycle continues when we look at how the disease is transmitted from an infected mosquito to an uninfected individual. So we call the mosquito the vector, but the disease, the Plasmodium species of the protist, is actually the pathogen that causes the disease. Let's move on to the next question. 1.3. How does immunization against disease such as diphtheria and polio limit the spread of these infectious disease? And guys, the key word is immunization. And from your discussions and lessons in class, if you remember that immunization basically involves introducing some form of a virus into an individual, and that form of the virus is generally a form of the virus that has been treated where the pathogenic parts have been removed. So if you think about the non-pathogenic parts, the outer protein coat, so that will be introduced into the host, into an individual, and the body responds by recognizing that non-pathogenic part of the virus as being a pathogen. It produces antibodies and it destroys it. And purely because the piece or the vector or the virulent part in that vaccine has been removed, and hence it does not elicit any immune response, or it does not produce any pathogenic effect on the body, and hence the body will produce the antibodies so that it can overcome or it can destroy the pathogen, and hence the body recognizes that in the future and it's able to produce more antibodies in response to that specific antigen on that vaccine. So let's look at the options. The answer here was D, and if you look at D, and the reason I'm going straight to this, guys, is because let me explain this answer clearly as it does not confuse us. Immunization reduces the multiplication of relevant pathogens in immunized host, and this reduces the chance of other people becoming infected. So the idea behind vaccination is that when you receive a vaccine, your body builds up the recognition of that, and that immunization reduces the multiplication of the pathogen in the body, simply because the body has built up immunity, it has built up the antibodies against that specific antigen on the virus, and hence it produces a quick response, thus preventing the individual from being able to get the symptoms of the disease, and hence infect other people, be it through contamination, through airborne, be it through saliva, and hence immunization. And some of the common diseases that we know that we immunize against are polio, measles, smallpox, and these are all that are caused by viruses. So the clue is that if a virus causes a disease, they should be treated with vaccines, and often not all viruses do have vaccines, which we must acknowledge, but viruses that have vaccines that have been created often are the ones that are better controlled in terms of the spread of infectious diseases. Very good to those that got this. Let's move on to the next question. Question number 4. The relationship between rhizobium and leguminous plants represents an example of. So guys, if we look at the options, parasitism, because rhizobium brings about the decay of legume when it dies, thereby returning to the soil. That's not true. It does not bring about the decay, so that's incorrect. Mutualism, because rhizobium denitrifies the legume and returns nitrogen gas to the atmosphere. That's not correct. Okay, that's incorrect. It's not that it's nitrates into the soil. Parasitism, because denitrifies the legume and returns nitrogen gas. It's not parasitism, guys. It is mutualism. And let's look at the last answer. So it's definitely mutualism, because it provides usable nitrogen for the legumes, and the leguminous plants provides the rhizobium with organic nutrients. And here we see the relationship between nitrogen fixing bacteria and the root nodules with the leguminous plants, where it becomes mutualistic, where the rhizobium benefits from the organic nutrients and the plant benefits from the ability of the rhizobium to fix atmospheric nitrogen and convert that into nitrates. And essentially that is how the relationship exists between the denitrifying rhizobium and the leguminous plants. So guys, we've reached the end of the first session. We've looked at some tricky multiple choice questions, and I know that it required a good discussion amongst yourselves. So let's have a short break, and then when we get back, we will look at a bit of terminology and some more lengthy questions. Welcome back, guys. I hope you enjoyed the break. Hope you're feeling a bit relaxed. I know there were some difficult questions there. Let's look at some terminology here. These are often very good to boost your confidence in the exam or in a test. So work with these, read the descriptions, and try and think of the closest answer that is to what the actual description fits. So let's go through these descriptions. So match each description in column A with the correct term for column B. Write the answer, the letter only, from column B in the space provided. So guys, what I tend to do with an activity like this is often I look at, there are two columns, you've got column A and you've got column B. And often column A are the long descriptions of the terms and column B are the answers. So what I often tend to do is I first want to read what column B is. I want to look at all these options here, so that as I get through and read through column A, some of this is going to be relevant, some of this is going to strike me and say, well, I know that I've just read the answer and this possibly could be the answers. So what is on offer in column B? Here we've got fungi, saprotrophs, archaea, hyphae, E, pneumonia, F, phytoplankton, G, AIDS, H, vector, I, eukaryote, J, plasmodia, and K, protozoa. So guys, I've looked at the various options that are available in terms of the column B. Let's look at the descriptions and try and match some of these descriptions to the terms listed in column B. So let's look at 2.1. Guys, long strands of a fungus that can extend over a substrate. Now what would that be? Remember, we looked at the terms. So guys, if you look at the structure of the fungus, we know that it's got these horizontal elongated threads, and these are called hyphae, and we know that here's the option that's given to us. So the answer here is hyphae, and that's D. Cool. An opportunistic infection of an HIV positive person. So guys, we know that when a person has AIDS, it's a result of the body's immune system becoming compromised. And this person is often, the immune system is weak to such an extent that the body now becomes susceptible to most of our common infections that the body normally can fight. However, having full-blown AIDS, the body's immune system is compromised, and hence a simple infection like common flu, pneumonia, TB are often fatal to a person that has got full-blown AIDS. So we're looking for an opportunistic infection that can attack an immune system when a person is extremely ill. And here we see the answer here. pneumonia. And if we go back up, guys, we know that a person that often has AIDS is most likely to be susceptible to diseases like infections like pneumonia, TB, and even the common flu, where the immune system does not respond to that infection. 2.3. Together with bacteria, fungi of this type break down dead organic matter. Guys, remember that fungi and bacteria can live off, some of them are heterotrophs, some of them are autotrophic. But however, there are some that cannot do either, and hence they rely on just simply producing enzymes that digest dead organic material, and hence they can absorb these. And we refer to these as your heterotrophs, your saprotrophs. And if we go down and look at possible answers for these, so I did see it coming up here. Here we've got saprotrophs, and so what we should be looking at is how these saprotrophs are able to convert dead organic matter and break them down and return those nutrients back into the soil. 2.4. An organism that causes malaria, and we just did that, guys. So what is the organism that causes malaria? We know that the mosquito is the vector, but the organism that causes malaria is Plasmodium, so the answer there is J. So you've got to read the question carefully. It's the organism that causes and not the vector that causes malaria. The kingdom of microbes which produce antibiotics. So what is it that produces antibiotics, guys? We know that when experiments were done initially, there were lots of experiments looking at how some of the yeast and some of rhizobium and some of the penicillin was used to treat infections. So these are all examples of fungi that produce a certain antibacterial substance that kills bacteria. So the answer there would be fungi, and in this case it's term A. Cells that have a membrane-bound nucleus and organelles. At the start of grade 11, guys, when your teachers looking at the characteristics of cells and in terms of your microorganisms, one of the first things that we had to differentiate was organisms or cells that have a true nucleus and those that do not have a true nucleus but have some kind of genetic material that is either a strand of DNA and RNA. And we refer to those two terms as either being eukaryotic cells having a true nucleus and prokaryotic cells having some evidence or strands of genetic material which could either be DNA or RNA. And in this case, guys, we're looking at cells that have a membrane-bound nucleus, so clearly a well-defined nucleus. And the term that describes these are your eukaryotes. So guys, your eukaryotic cells are cells that have a distinct nucleus that is bound by a membrane. And lastly, we're looking at animal-like protista. And guys, if we look at animal-like protista, we know that your protozoans are the animal-like protista, and that's the option K. So I hope this was a good opportunity for you to look at what we've done through the term, reflect on some of these important terminologies. You need to build up a vocab list of these important terms for each section and use these to revise and to familiarize them yourselves, especially looking into an assessment where terminology is essential to establishing the basics of the topic. Let's move on to some longer questions. Guys, here the next question's got some illustrations of different invertebrates that we possibly looked at. Some of them are belonging to the kingdom so we've got an illustration here of Paramecium species, guys. Again, it belongs to protist. We've got a very nice diagram here of an amoeba. Again, we're seeing an animal-like protist which has got pseudopodia, which is characterized by those folds and those invaginations on the outer cell wall, cell membrane. And the last one is an illustration of a Euglenia, which is a freshwater species. And what we need to look out for is the presence of chloroplast here and some storage vacuoles here, which are quite characteristic of an organism that has the ability to photosynthesize and store some of those nutrients or some of that into these pyrenoid vesicles or structures. We also see the presence of a flagellum or a tail-like structure in this specific Euglena species. So guys, let's get to these questions. We're going to read through these questions very quickly and then we're going to try and work out some of these answers. To what kingdom do these organisms belong? So we're looking at all three organisms and as I was going through identifying them, you'd have realized that I had to sort of contextualize and put these organisms into the context of where they are from and what do they represent. So it's very good that at the onset, when you see an illustration of a microorganism, try and identify whether it is a bacteria, whether it's a virus, protist, or fungi. And that helps to then try and contextualize its characteristics based on knowing which group it belongs to. And that's essentially very important at the onset of any question where you've got illustrations of your invertebrates, especially the microorganisms. Which of these organisms are autotrophic? Give a visible reason to support your answer. Guys, we all know that autotrophs are organisms that are capable of manufacturing their food, but we need to look at the diagram and give a visible reason to support your answer, and hence it needs to come from the source that is provided. 3.3. Which organism could be considered as being intermediate between plants and animals? Give one reason, give one visible reason, again, focus on the word visible, that might suggest that it is plant-like, and one reason why it is animal-like. So two parts to the question where we've got to give reasons as to why it's plant-like and animal-like with both evidence from the diagram. 3.4. What type of food would be stored in the pyrenoids? Explain your answer. So guys, I've read through these questions. I'm going to give you guys probably 3 minutes to work through these, and during those 3 minutes I'm going to flip through these questions, and then obviously after a minute I'll get through the diagrams so that you can see these diagrams as you work through this. So 3 minutes, guys. Read through these questions, look at the diagrams, let's come up with some good answers, have some good discussions with your friends. Welcome back, guys. I hope you guys had fun. So the trick behind this question was that you needed to look at some of these diagrams and then relate your answers to using evidence from the diagrams. So as you guys were working through that, I showed you the questions, I tried to show you some of the diagrams and then go back onto the questions, hoping that you would be able to be familiar with those questions and obviously look for the evidence in the diagrams to support your answers. So let's look at these questions, some quite interesting questions that we need to work through and obviously find the evidence from the question on this. So again, a very quick reflection going through these diagrams. So we saw that we've got a paramecium, we've got an amoeba species, and we've got Euglena. So let's look at, to what kingdom do these plants belong? Or do these, organisms belong? And that's the key word. Which is the kingdom that we're looking at? So guys, the kingdom we're looking at was, okay, so I'm just going to click on my answer here, and that is Protista. So that was the third group of organisms that we've looked at. So the first group we looked at the start of the year was viruses, then we went on to bacteria, we saw them becoming more specialized, more evolved, and then finally we looked at Protista, where we saw that there were animal-like protists and plant-like protists. The animal protists probably had cellular structures that allowed them to move, like the paramecium, and they had flagella which allowed them to move, but the plant-like protist had some important structure which we see for the first time, which was chloroplast, and they had the ability to be able to photosynthesize as well. So well done to you if you've got 3.2 correct, 3.1 correct. Let's look at 3.2. Which of these organisms are autotrophic? Give a visible reason to support your answer. And guys, if you look at the diagram, if we go back to Euglena, you'll see that it showed the presence of chloroplast in there. So here's the Euglena diagram, guys, for you to very quickly refer to. So I'll repeat the question. Which of these organisms are autotrophic? Give a visible reason. And in all of these, guys, oops, so let me just get my pen out before this. Okay, we see the presence of chloroplast here. If we go back to the others, guys, no distinct chloroplast in this, and hence cannot photosynthesize or autotrophic. And in this paramecium, we see the absence of any specific chloroplast or organelle that's capable of photosynthesizing. And hence, of the three, it's the Euglena that is the organism that is able to photosynthesize, and the evidence from the diagram is that it shows the presence of chloroplast in the organelle. Well done to you if you got that one correct. Let's look at 3.3. Which organism could be considered as being the intermediate between plants and animals? Here we've got to decide on the organism first. That's the first part of the question, and we've got to give one reason as to why we suggest that it is plant-like, and another reason why it's animal-like. So guys, let's get through this. So we're looking for characteristics of both plant-like and animal. And voila, we're looking at Euglena, and he seems to be, or she seems to be, the choice of today. And what are the characteristics that are present? So we're seeing that it showed a distinct flagellum and the evidence of chloroplast in the plants, in the cell. And hence, it has both animal-like characteristics by the presence of the flagellum, and it has plant-like characteristics by the presence of chloroplast being in him. So well done if you got that correct. Let's look at the fourth question. Cool. What type of food would be stored in the pyrenoids? Explain your answer. So I'm going to very quickly go back to our reference point, the diagram, guys. And if we look at this organelle, this organism, guys, it has this organelle which is chloroplast. And you guys can recollect from grade 10, you've done the process of photosynthesis. In an organism that does photosynthesize, it needs to store the products of photosynthesis in its cytoplasm. And in this case, the chloroplast does produce starch, it does produce glucose, and it stores that glucose in the form of stored food. In this case, it's going to be starch, guys, in the form of those pyrenoid granules. So we're looking at the answer for this question was that it's the starch which is present in an osmotically inactive form and hence does not affect the ability of the organism to retain water or to lose water. So guys, I want you guys to have a short break, stretch a bit, and then we get into the last segment where we work with some more interesting questions on immunology. Welcome back, grade 11 learners. Let's look at the final segment. Here we're looking at some interesting questions on immunology. And guys, to put this into context, we've looked at vaccines and how vaccines produce an immune response in the body so that the body then builds up immunity against specific antigens. This question deals exactly with that. But here we're looking at how antigens bring about an immune response, and we look at how immunity can be established by using the understanding behind how antigens and antibodies work. So here's a question that deals with a hypothetical example of a student carrying out an experiment. 4.1. Antigens exist on cell surface. So we know that all cells have certain surface receptors that are called antigens, which the body recognizes between self and non-self. Those that are recognized as non-self become pathogenic, and hence the body produces some kind of immune response against it. So the following diagrams show different forms of antigens and their relative distribution found on the surface of different strains of Staphylococcus bacteria. So here we've got different strains of the Staphylococcus bacteria, quite a tongue twister. Here we've got strain M, N, P, Q, R. And what you'd notice that, guys, on the surface of these different strains are different shaped antigens. And essentially what this illustration is showing is that the antigens have specific shapes which are unique, and hence when the body responds to producing immunity, the body needs to recognize these surface antigens as being unique and different in order for the body to produce an immune response. So let's read further into the question. You wish to make a single vaccine that will be effective against as many strains as possible, shown by this bacteria. So essentially the aim of this task is, let's develop a vaccine that can recognize as many strains as possible so that we can administer a single vaccine dose that can be effective in reducing or being effective against the different strains from M all the way up to R. So let's read the questions, and then I'm obviously going to give you some time to work through this. Which strain would you use to make the vaccine? 4.1.2, explain your answer to the question. So guys, essentially what I'm going to leave you with in the next 2 minutes is look at the diagram, look at the surface antigens on these different strains, and identify the one that has all or most of the antigens, and you will need to decide on which one has them and how you would use them best in terms of producing an antigen. And you've got to explain that in the next part of the question. So guys, 2 minutes, have a quick discussion with your friends, and let's get back and we'll look at the answers. Welcome back, guys. I can sense that you guys had a good discussion. I wish I was there with you now to listen or listen as to what you guys discussed. I think the clue to this question was to look at the different strains and to see which of these strains had the most number of antigens and whether it had all of them in comparison to the rest of the strains. So again, I'm going to read the question so that we can look at the answer correctly. Okay, so which strain would you use to make the vaccine? And guys, if you looked at the different strains, if you looked at strain M, it's got 1, 2, 3 different surface antigens. If you look at strain M, N, it's got just 2. If you look at strain P, it's got 1, 2, 3. Again, not all. Strain 4 has got a unique, 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 and unique. So strain Q has got 4 unique surface antigens. Let's look at strain R. Strain R has got 1, 2, so it's got 2. So if we look at the different strains, the strain that has the most number of surface antigens would be the one that I would choose. And looking at this, it's going to be Q. And that's possibly the explanation that you need to have for the next question. So the correct answer would be strain Q, as I said there, and that's my reasoning. But let's put that answer down in terms of a logical explanation to the question. Okay, I'm just going to read that question again for the benefit of you all. Explain your answer to that previous question. Okay, so we've got to explain, guys. So let's look at this. So remember, we counted out the surface antigens. Definitely it has four different antigens, which would allow it to be the most effective against all the strains. And if you decide to use M, I just want to go back to M and I'll tell you why M. If you look at M, it's got, although it seems to have quite more than the other N and R, it only has 3, and hence you cannot use M as it has no surface antigens to match R, and hence you cannot use M. Okay, so that does make sense as to why you would use strain Q. So guys, let's look at some more questions related to immunity. Students were asked to draw an antibody that would be effective against strain P. The following diagrams were presented by 3 students. Which student has drawn the most effective antibody against strain P? Explain your answer. So guys, we need to definitely go back to the strains. And if we look at strain P, guys, this is what we're looking at. We're looking at this. So if you look at strain P, it's got 3 different surface antigens on them. And these are a semicircle, a solid block, and then obviously an arrow on it. So in the context of this, we need to develop an antibody to be able to identify that. So let's get straight to my question. And let me see, I've got the answer on the next page. So guys, if you look at why we would do this, the answer for this would be, we've got to look at an antigen on P that will fit into these complementary shapes that we see here. And if you look at P, guys, the answer, let's go to the answer first, and then I'm going to explain to you why we've decided on that. And the answer here is student Y. And the reason for that is that the antibody has 2 different shapes, allowing it to match up with 2 of the antigens on P. And let's go back to that. I know that we're going back and forth, but I think it's important to relate that to the diagram. If you look at, it's got 2 different antigens, and if you look at the shape of that, this is definitely going to fit into that shape on the antibody, and hence the explanation as to why we would choose P as the correct molecule, why we would choose student Y as the correct antibody for that specific strain P. Okay, cool. So guys, we looked at those questions. Let's look at one more related to this. T lymphocytes play an important role in the immune system. Name and explain the function of each type of T lymphocyte. So guys, remember that when we talk of HIV and how the body responds to an HIV infection, we refer to our CD4 cells, our complementary defense cells, which are part of your T lymphocytes. So we've got to discuss the role in the immune system, and we've got to name and explain the functions of each type of lymphocyte. So guys, I'm going to get straight into my solution. I'm going to read that out, and I hope that you guys can understand the answer and take this into context of how the body fights off infections in terms of when a person is susceptible to an infection. So again, remember we discussed your CD4 cells, and these are your T helper cells or your CD4 cells, and these start the immune response to an infection. So these basically will then respond to the body being infected. Your killer T cells, which are a different type of T cells, they destroy the body cells infected by the virus and parasites. So often remember that viruses infect healthy cells, and these viruses will then invade the nucleus and trigger the process of DNA replication or viral replication within these host cells. The T cells will destroy these body cells that have been infected with the virus. Then you have a group of cells called suppressor cells, and these stop the immune response once the infection is over. And often, guys, there is identification, there is the immune response, and then there is the weaning off the immune response, because your body cannot constantly produce soldiers or antibodies against a specific infection. So the first part of the immune response is to recognize, identify. The second part is to destroy, and that is done with your killer T cells, as I explained there. And finally, that immune response cannot be one that the body constantly produces, and hence you need to have a parasympathetic response where it stops the immune response, it stops producing the antibodies, and hence the antibody count of your T helper cells or your T killer cells reduces, and then hence we refer to these as your suppressor cells that stops the immune response once the infection is over, so that your body does not constantly waste energy on producing an immune response against something that has already been treated or has been destroyed. Cool. So well done to you if you've worked it out. Let's look at the last question, and we're going to read through this, guys, and I'm going to give you an opportunity to have a discussion, but probably we might not have time for that, so I'm going to read through this and maybe get to some of the questions. Nitrate, a common ingredient in fertilizer, is found in surface runoff from agricultural land that makes its way into streams, lakes, coastal zone, and is leading to the cause of degraded water quality worldwide. And we see this throughout South Africa, we see it around the world. A scientist, Bradley Cardindale, at the University of Michigan, investigated how useful algae were at cleaning up pollutants in water. Algae are able to absorb the nitrates from the water, thereby decreasing the level. He wanted to investigate whether water that contains a rich mixture of species of algae is better at removing nitrate than water with only a single species. So what he was looking at was he wanted to compare a mixture of algae in treating nitrates as compared to a mixture with a single species. Cardinal set up an experiment where he discovered that the greater the number of different species of algae present, the less the nutrient pollution there was in the water. So here we're seeing that he set up an experiment where he was able to successfully prove that the greater the number of species of algae in polluted water, the less the pollution was of that water, or the algae were able to remove the nitrates from the water. So it's quite a lengthy paragraph to read through, but I think it's important that you read through, underline, circle keywords in it. So guys, I'm going to read through these questions. We might not have time to work through all of them, but I think you guys can engage with as many as we can. From the explanation, draw a sketch graph to represent the findings of Cardinal. What was the hypothesis of the experiment? 5.3. Name another group of organisms that can be used to clean up pollutants in water, such as sewage. And 5.4. Other than being able to absorb certain pollutants, explain why algae play such an important role in an ecosystem. So guys, let's get straight to the answers. I hope that you guys are having a discussion whilst I'm going through these solutions. So the first one, from the explanation, draw a sketch graph. And guys, a sketch graph is essentially drawing an interpretation of what the possible trend of this graph would be. And from that you can see that here the independent variable would be the number of species, and hence you're going to get a mark for putting that on the x-axis, and the dependent variable would be the nitrate content of the water. And essentially from that last line in the paragraph, it clearly showed that as the number of species of algae increased, the amount of nitrates decreased, and hence we referred to this as an inversely proportional relationship, and hence we will indicate that as the line decreases as the number of species of algae increase. So well done if you could get that answer correct. Let's look at the next question. What was the hypothesis for this experiment? Guys, remember that the hypothesis is a statement, it's an educated guess. You need to be specific that you include both the variables in your hypothesis. That includes the dependent and the independent variable, and let it be a statement of intent and not an investigative question. So in this case, the hypothesis would be a greater number of species of algae will reduce most of the pollution of nitrates in fresh water. Let's look at 5.3. Name another group of organisms that can be used to clean up pollutants in water, such as sewage. So guys, we know that bacteria can be used in bioremediation, so they're quite useful that way. Guys, we've come to the end of the session. We've run out of time, but I'm glad that you've enjoyed the lesson. I hope that you've interacted with your colleagues. Study hard, work smart, use some of these revision in your studies. All the best, take care.